A signal peptide is a short peptide present at the N-terminus of the majority of newly synthesized proteins that are destined towards the secretory pathway. These proteins include those that reside either inside certain organelles, secreted from the cell, or inserted into most cellular membranes. Although most type I membrane-bound proteins have signal peptides, the majority of type II and multi-spanning membrane-bound proteins are targeted to the secretory pathway by their first transmembrane domain, which biochemically resembles a signal sequence except that it is not cleaved. Translocation. In prokaryotes, signal peptides direct the newly synthesized protein to the Secretory protein conducting channel, which is present in the plasma membrane. A homologous system exists in eukaryotes, where the signal peptide directs the newly synthesized protein to the Sec61 channel, which shares structural and sequence homology with Secretory, but is present in the endoplasmic reticulum. Both the Secretory and Sec61 channels are commonly referred to as the translocation, and transit through this channel is known as translocation. While secreted proteins are threaded through the channel, transmembrane domains may diffuse across a lateral gate in the translocation to partition into the surrounding membrane. Signal peptide structure. The core of the signal peptide contains a long stretch of hydrophobic amino acids that has a tendency to form a single alpha helix. In addition, many signal peptides begin with a short positively charged stretch of amino acids, which may help to enforce proper topology of the polypeptide during translocation by what is known as the positive inside rule. At the end of the signal peptide there is typically a stretch of amino acids that is recognized and cleaved by signal peptidase. However, this cleavage site is absent from transmembrane domains that serve as signal peptides, which are sometimes referred to as signal anchor sequences. Signal peptidase may cleave either during or after completion of translocation to generate a free signal peptide and a mature protein. The free signal peptides are then digested by specific proteases. Cotranslational versus post-translational translocation. In both prokaryotes and eukaryotes, signal sequences may act cotranslationally or post-translationally. The cotranslational pathway is initiated when the signal peptide emerges from the ribosome and is recognized by the signal recognition particle. SRP then halts further translation and directs the signal sequence ribosome mRNA complex to the SRP receptor, which is present on the surface of either the plasma membrane or the ER. Once membrane targeting is completed, the signal sequence is inserted into the translocation. Ribosomes are then physically docked onto the cytoplasmic face of the translocation and protein synthesis resumes. The post-translational pathway is initiated after protein synthesis is completed. In prokaryotes, the signal sequence of post-translational substrates is recognized by the Sec-B chaperone protein that transfers the protein to the Sec-A ATPase, which in turn pumps the protein through the translocation. Although post-translational translocation is known to occur in eukaryotes, it is poorly understood. It is however known that in yeast post-translational translocation requires the translocation and two additional membrane-bound proteins, Sec62 and Sec63. Signal peptides determine secretion efficiency. Signal peptides are extremely heterogeneous and many prokaryotic and eukaryotic signal peptides are functionally interchangeable even between different species. However, the efficiency of protein secretion is strongly determined by the signal peptide. Nucleotide level features. In vertebrates, the region of the mRNA that codes for the signal peptide can function as an RNA element with specific activities. SSCRs promote nuclear mRNA export and the proper localization to the surface of the endoplasmic reticulum. In addition, SSCRs have specific sequence features. They have low adenine content, are enriched in certain motifs, and tend to be present in the first exon at a frequency that is higher than expected. See also, protein targeting, target peptide, topogenic sequence, references, Blabel-G, Doberstein-B transfer of proteins across membranes. Presence of proteolytically processed and unprocessed nascent immunoglobulin light chains on membrane-bound ribosomes of marine myeloma. J. Cell Bio 67, 835 €51 doi, 10.1083/JCB 67.3.835. PMCA 2,111,658. PMIDA 811,671. Rappaport T protein translocation across the eukaryotic endoplasmic reticulum and bacterial plasma membranes. Nature 450, 663 Euro 9 doi, 10.1038/Nature 06384. PMIDA 18,046,402. Von Heij, G. Gavel, Y topogenic signals and integral membrane proteins. J. Biochemically 174, 671 Euro 8 doi, 10.1111 per joule 1432-1033.1988 TB 14150X. PMIDA 3,134,198. Walter P. Ibrahimi I. Golgi translocation of proteins across the endoplasmic reticulum. I-signal recognition protein binds to in vitro assembled polysome synthesizing secretory protein. JCB 91, 545 Euro 50 doi, 10.1083/JCB 91.2.545. PMCA 2,111,968. PMIDA 7,309,795. Gilmore R., Golgi, Walter P. protein translocation across the endoplasmic reticulum. I detection in the microsomal membrane of a receptor for the signal recognition particle. JCB 95, 463 Euro 9 doi, 10.1083/JCB 95.2.463. PMCA 2,112,970. PMIDA 6,292,235. Gar paragraph RLICHD, Pranes, Hartmann E., Kayles KU, Rappaport TA. A mammalian homologue of SEC61P insect is associated with ribosomes and nascent polypeptides during translocation. Cell 71, 489 Euro 503 doi, 10.1016/0092-8674, 92, 90517 G. PMIDA 1,423,609. Panzer, S. Dreyer, L. Hartmann, E. Kostka, S. Rappaport, T.A. Post-translational protein transport in yeast reconstituted with a purified complex of sec proteins and Ca2p. Cell 81, 561 Euro 570 doi, 10.1016/0092-8674, 95, 90077-2. ISNA 0092-8674. PMIDA 7,758,110. Kobe L., Z.C., Bao J. Optimize signal peptides for the development of high-expressing CHO cell lines. Biotechnol. Biong. 110, 1164 Euro 73 doi, 10.1002/bit 24776. PMIDA 23,124,363. Von Heij G. Signal sequences, the limits of variation. J. Mobile 184, 99 Euro 105 doi, 10.1016/0022-2836, 85, 90046-4. PMIDA 4,032,478. Palazzo, Alexander F. Springer, Michael. Shibata, Yoko. Li, Chuang-Shang. Diaz, Arnosha P. Rappaport, Tom. The signal sequence coding region promotes nuclear export of mRNA. PLOS Biology 5, E322 doi, 10.1371/journal PBIO. 00503222. ISNA 1544-9173. PMCA 2,100,149. PMIDA 18,052,610. Senek, Can. Qu. Honorable Nian. Zhang, Hui. Tanorski, Stefan P. Ekf, Abdullah. Dirty, Adnan. Tarzan, Murat. Moore, Melissa J. Palazzo, Alexander F. Raff, Frederick P. Genome analysis reveals interplay between 5 UTR introns and nuclear mRNA export for secretory and mitochondrial genes. In Snyder, Michael. PLOS Genetics 7, E10013666 doi, 10.1371/Journal PGEN. 1,001,366. ISNA 1553-7404. PMCA 3,077,370. PMIDA 21,533,221. External links. Signal peptide at the U.S. National Library of Medicine. Medical subject headings, SPDB, Signal P predicts the presence and location of signal peptide cleavage sites in amino acid sequences from different organisms.